Are live now. Hello, everybody, and welcome to the August 2016 micro seminar. We're very honored to have Derek Lovley from the University of Massachusetts Amherst with us today, describing some of his work with electroactive bacteria and how they are interacting with each other. Before we get started, I just want to remind everyone that we will be taking questions at the end, and tweet your questions all the way through the seminar to the hashtag #USeminar or directly to the handle @microseminar, and I'll make sure to get those questions to Derek. So without any further ado, Derek, take it away.
Thank you, Cameron. It's a real pleasure to have this opportunity. Let me switch to hopefully I'll get this right now. Okay, do you see my presentation now?
That's perfect.
Perfect, great. So yeah, so this morning I'd like to give a brief overview of some of our recent studies where we've been looking at this process known as direct interspecies electron transfer, which is a relatively recently discovered mechanism for microorganisms to exchange electrons in anaerobic environments. So this was a process that was first discovered in studies conducted by Zara Summers when she was a PhD student in my laboratory, and I'll go through how she made this discovery and then talk about in more detail or depth a diet-based community that we investigated for a number of years, and this is anaerobic digesters treating brewery waste. And with this community, we've been able to go into a fair amount of detail, starting out with metagenomic and metatranscriptomic studies, and then developing synthetic defined communities to study the process going on in the digesters further, and going down several levels to looking at individual microbes and then individual proteins and their structure and function. So one reason that there's interest in this is it's a very applied application of diet. We feel, and I guess our funders feel that studying diet is useful for also other practical processes. One would be, for example, microbial electrosynthesis, where microbes, instead of accepting electrons from another cell, they're using a fairly similar process to accept electrons from an electrode to reduce carbon dioxide, to produce fuels and other commodities. And another practical spin-off. Okay, my presentation has just completely frozen.
Okay, let's see. Why don't you drop out of the screen share and just try redoing that part?
Yep. Yeah, except I can't find my cursor. This is really embarrassing.
Oh, that's okay.
Any suggestions, other ways to get out of this?
Let's see. Does the escape button work?
Boy, I'm very sorry about this. It's frozen and all right.
That's okay.
I guess we can go nuclear and just shut it off and start over.
Yeah, maybe the best thing is if you just quit out of your browser and then restart your browser and re-click on the link. We'll still be here. I'll hold the fort for a second while you're gone, and we'll restart.
Can you still hear me?
Yes, I can.
Yeah, I even tried shutting it down and clicked the shut down, and it won't shut down. So it's just, Unbelievable.
Okay, let's see. Are you on a Mac?
Yeah.
Oh, there you go. Looks like you popped out there.
Yeah, I did.
Okay, can you restart your presentation to see how that works?
Sure. Yeah, the presentation's got a horrible. Let's. Okay, can you see it again?
I can.
Yeah. All right. I'll skip ahead to where I was. Okay, so I was talking about microbial electrosynthesis, and then another practical spin-off of this extracellular electron exchange has been these electrically conductive pili that are important in the process, and this is actually a big part of the focus of my lab now, is synthetic biology on those for making new types of electrically conductive materials. Paper recently just came out on that. And I'll talk a little bit about that at the end. But what I'd like to primarily focus on are these anaerobic microbial communities that share electrons. And to point out, just remind everybody, so we're starting at the same place. Typically in these anaerobic environments, especially those where methane is being produced, there's no one microorganism that can take a fermentable organic substrate all the way to terminal products. Instead, there are microbial food chains that cooperate to carry out that process. And there's basically a successive oxidation of the organic substrates until you finally convert the fermentable organic compounds to methane. So this is the simplest model you could possibly draw for converting a fermentable compound to methane. So where the fermentable compound is attacked by a fermentative organism, produces acetate and carbon dioxide. The acetate is converted to methane by an acetoclastic methanogen. This also results in the release of electrons. And of course, it's not thermodynamically possible to just release electrons into water. Those cannot exist. So the microorganisms have to come up with a strategy for disposing of those electrons. And the common microbial solution is to use those electrons to reduce protons and form hydrogen gas. And this works out well in these communities because hydrogen is a high energy substrate that in methanogenic environment is consumed with the reduction of carbon dioxide to methane by the hydrogenotropic methanogens. So this is a process that's been termed interspecies hydrogen transfer. It's been known about for about 50 years. It was discovered at the University of Illinois by Bryant et al. And this was discovered in the study of what was initially thought to be a pure culture, but it turned out to, in fact, contain two microorganisms. One was the methanogen, which can consume hydrogen with the reduction of carbon dioxide to methane, and the other organism was what was called the S organism. The S organism metabolizes ethanol to acetate with the reduction of protons to hydrogen gas. So this consortium was converting hydrogen or ethanol to methane with interspecies hydrogen transfer. This concept then of interspecies hydrogen transfer has now been expanded over the years, primarily in studies originating out of the Bryant lab to include beyond ethanol a variety of volatile fatty acids, long-chain fatty acids, and even some aromatic compounds. And this has been the model for how fermentable compounds can be converted to methane in a diversity of methanogenic environments. So we were interested in interspecies hydrogen transfer, and but the S organism is no longer available in culture. So the modern day analog of the S organism is Pelobacter carbonolicus. This is a microbe that was discovered by Bernard Schenck, and it has that same metabolism of utilizing ethanol as the electron donor and reducing protons to hydrogen gas. And we were particularly interested in that Pelobacter because they're closely related by phylogeny and 16S sequence to various Geobacter species that we've been studying for some time. And when you look at the genomes of these various organisms, it was very apparent that the more fermentative type metabolism of the Pelobacter had evolved from an ancestor which was more like the modern day Geobacters, which have the respiratory metabolism. So in order to understand better how this syntrophy might evolve with an organism like Pelobacter, we started with two geobacter species, sulfur-reducens and metalloreducens, that are closely related to the Pelobacter species. And we put them in a medium in which neither of them could grow independently. Ethanol was the electron donor, and geobacter metalloreducens can convert ethanol to acetate, but we provided only fumarate as an electron acceptor. Metalloreducens cannot reduce fumarate, whereas sulfur-reducens cannot metabolize ethanol, but it can use fumarate as an electron acceptor, reducing fumarate to succinate. It's also important to point out that when sulfur-reducens is growing on fumarate, it does not normally express a large abundance of pili, and the initial result was that the two microbes didn't communicate. They just sat there. The co-culture didn't grow, but after a couple of weeks, growth did start. And when we look back and investigated this further, this metabolism of the co-culture starting was associated with a one base pair change in one regulatory gene in geobacter sulfur-reducens. This introduced a stop codon into the regulatory protein, the gene for the regulatory protein, PIL-R. And this had really dramatic results on geobacter sulfur-reducens. It began expressing abundant pili, and then very importantly, not only were the pili produced, but also there was a large abundance of a multi-heme C-type cytochrome known as OMCS, which is specifically localized on the pili. And that upper left-hand panel is showing the TM image of the strain with the mutation. You can see the abundant pili, and everywhere there's a uniform black dot, there's an OMCS, as this was stained with a gold-labeled antibody procedure. As shown in the lower left-hand panel, there was just a lot by Western blot. You can see there's just a lot more OMCS once that mutation took place. And associated with that mutation and the growth of the co-culture was the formation of these large aggregates, sometimes a millimeter across. As shown on the left here, they're red because of the abundant C-type cytochromes in both geobacter species. Here's a fish image. Shows that basically the two organisms are about in equal abundance. When you look at this with transmission electron microscopy, you can see that between the cells there's this incredibly dense network of the pili of geobacter, which we know are electrically conductive. Here's another look at it through a thin section through one of the aggregates, so you can see the cells. You don't really see pili that well in thin section, but this was also immunogold labeled, and you can see the OMCS throughout the matrix. The metalloreducens was also expressing pili at very high levels, and also genes for extracellular C-type cytochromes were also upregulated. So both cells are producing a lot of conductive pili, a lot of outer surface C-type cytochromes. When we look at the transcriptome of this co-culture, you could see that there was really low expression of genes in geobacter sulfur-reducens for uptake of hydrogen or formate, which can sometimes serve as a substitute for hydrogen in interspecies electron transfer, suggesting that hydrogen and formate were not important intermediates. And also, we built a strain of geobacter sulfur-reducens that could not use hydrogen or formate. And when we started the co-cultures with that strain, they grew just as well as starting the strain with wild type. So this indicated to us it was probably the direct interspecies electron transfer, which we'd actually speculated a long time ago might be possible when we first discovered that geobacter produced electrically conductive pili. So I want to talk about these pili for a couple minutes. So this is a transmission electron micrograph, which shows geobacter stained in red, and hopefully you can see against the blue background this network of white wires. These are the filaments that geobacter produces that are electrically conductive. They have homology to type 4 pili. So they're about 3 nanometers in diameter and can range from up to 10 to 20, maybe even 30 microns in length. So just to go over some of the key points about these electrically conductive pili, which I'm primarily going to call E-pili, I think, in this talk, but you'll see for many of the citations, we used to call these microbial nanowires. So the reason we're changing the name is that this microbial nanowire term has been co-opted by a number of other investigators to now describe all kinds of filaments, some of them of unknown composition, or it's not clear whether they're conductive under physiologically relevant conditions. Probably the best known are the microbial nanowires, in quotes, of Shewanella oneidensis, which turn out not to be pili, but are in fact lipid-based extensions. And they are, although you may see in reviews that these Shewanella nanowires are involved in interspecies electron transfer, that's just a mistake. That actually has never been shown. So I'm going to use this term E-pili, and one thing to recognize is that this long-range electron transport through a biological protein over micrometers distance is a novel concept in biology. Nothing like that's ever been seen. And our original thought was, well, this geobacter evolved these primarily to grow on insoluble electron acceptors like iron oxides. So this is a scanning electron micrograph showing geobacter colored in green, and it's growing on a poorly crystalline iron oxide that was synthesized in the laboratory in such a way to try to replicate the iron oxides that geobacter typically uses in soils and sediments. So what geobacter is doing, of course, it's oxidizing organic compounds to carbon dioxide within the cell, and then it faces this physiological challenge of getting electrons outside the cell onto the insoluble iron oxide, where it can then reduce the ferric iron to ferrous iron. So in some of the early studies of geobacter growing on iron oxide, Susan Childers, who is a postdoc in my lab, noticed that geobacter would express filaments when grown on iron oxide, but not soluble electron acceptors, such as chelated iron. If you see on the left, these big filaments are actually flagella, but if you look closer, here are the pili. And then on the right-hand side is a higher magnification. Again, there's the pili, about 3 nanometers in diameter. And these pili do have homology to type 4 pili found in other microorganisms, and like those other pili, they're primarily produced from a monomer known as pil A, which gets assembled into the cell, to produce the filament. So another important observation was made by Gemma Reguera. She noted that under some conditions, as shown here, in transmission electron microscopy, that iron oxides would specifically bind to the pili. So this was an interesting observation, because if the iron oxide is way out here, the question is, well, how are the electrons generated from central metabolism getting out to the iron oxides? And the simplest explanation for that was that there's some mechanism for electron transport along those pili. And in the initial studies, Gemma worked with Kevin McCarthy, who was a PhD student in physics, to use conducting tip atomic force microscopy to measure the conductivity just across the diameter of the pili, but these were clearly conductive at least across their diameter, which gave us enough reason to speculate with all the other data that there's probably electron transport along the length, and that these could function as what we call microbial nanowires. It's taken some time in order to get measurements of electrical conduction along the length of individual pili. We just this year, published this result where you can throw an pili down on a microelectrode array and measure the conductivity of individual pili. You get this ohmic response. And the important observation is that not only are they conductive in cytochrome-free regions of the pili, but also the conductivity of these pili under physiologically relevant conditions is more than sufficient to account for the rates that geobacter transfers electrons outside the cell onto extracellular electron acceptors like iron, other cells, or electrodes. Okay, another important point about the E-pili is that they have a type of conductivity that has never been seen in a biological protein, and this is metallic-like conductivity. So in typical biological electron transport, of course, you have discrete redox carriers, and if those carriers get close enough, typically less than 2 nanometers, the electrons can hop or tunnel from one molecule to another. Metallic conductivity is substantially different. Here, the electrons are delocalized along the material, and so if there's a potential difference across, in this case, the filament, there'll be electron flow in that direction. So it's really important to recognize, though, that metallic-like is referring to the mechanism of conduction. It does not mean that the pili have a conductivity that's equivalent to that of metals. Sometimes you might read in papers that if the pili are so conductive like a metal, why doesn't it just drain all the electrons out of geobacter and have a really rapid electron transfer? The fact is that the conductivity, as I'm showing here, is well below that of metals, and somewhere in between that of various synthetic conducting organic polymers. Okay. In addition to conduction along the length, if you have networks of pili, you can get long-range electron conduction, now up to centimeters in length, as was shown by studies by Mikhail Malvankar. And one of the early observations that Mikhail made was that he was a physicist, so the first thing he did was look at temperature optimum, and as he decreased the temperature, note this is a log scale over here on the y-axis, he saw an exponential increase in the conductivity of those pili networks. So that's a really unexpected result for a biological protein. If you have electron hopping, as you decrease the temperature, the conductivity should decrease. So he was actually seeing the opposite, a dramatic increase, but to physicists, this really wasn't, it's been seen before, and Mikhail knew right away that this is a response that's seen in other materials, such as synthetic organic conducting materials, such as polyaniline, where you get the same type of temperature response. Another important observation early on was the pH response. Again, log scale on the y-axis, and note that with decreasing pH, there's a dramatic increase in the conductivity. In fact, the conductivity is highest at pH 2. This is a pH which certainly denatures any cytochromes associated with the pili, but the pili themselves are very robust and highly conductive. And this pH response has recently been further confirmed with individual pili. So this suggested that the conductivity in the pili was similar to that in these synthetic conducting organic polymers, and that it was a metallic-like conductivity, which was surprising because just before Mikhail's discovery, there was a number of very authoritative reviews that said there'll never be metallic-like conductivity in a biological protein. So it's really important for us to try to understand how there could be biological or metallic-like conductivity in these pili. And one thing that's known for the synthetic organic materials is that their metallic conductivity results from overlapping pi-pi orbitals of aromatic compounds. So Mikhail looked at this and initially through standard X-ray diffraction saw evidence for the overlapping pi-pi orbitals, and then did more sophisticated synchrotron and rocking curve X-ray diffraction, which also revealed that feature. An important point, as can be seen in the lower right, is that the pH 2, there was a much greater abundance of the pi-pi stacking than at pH 10.5, and that's consistent with the much higher conductivity at that low pH. Another important observation is this lack of parent pi-pi stacking in pili from a strain known as Aero5. And this is a strain that we genetically engineered to produce pili that are not electrically conductive. And the way we generated this was by swapping in an alanine for the 5 aromatic amino acids at the carboxyl end of that pili monomer for the pili. And we were really lucky. We got a strain that is shown here, and TEM still produced pili. And not only that, I'm showing here immunogold labeling for the OMCS. So the cell is, even though we've changed the pili sequence, still making pili that look like a wild type and still properly expressing the outer surface cytochromes. Yet the conductivity of these pili is much lower. And this was shown first in analysis of networks, and then as we got able to measure the single filaments, this is also evident in individual pili as well. So these aromatic amino acids are responsible for much of the electron transport through the pili. And this has a dramatic impact on extracellular electron transfer. For example, here of the Aero5 strain, it's shown that it cannot effectively reduce iron oxide when compared to the wild type. Another way to look at metallic-like conductivity is something called electrostatic force microscopy. So this is a technique that's been used to investigate the charge propagation in carbon nanotubes, as shown on the left. If you inject charge into a carbon nanotube, the whole tube will light up due to the charge propagation. In the right-hand panel are results of Mikkel's working with Ebru Yeltsin, where they did electrostatic force microscopy on the pili. Injected charge is shown by the red arrow, and you can see the whole pili lights up. In this case, there was another pili contacting the pili to which charge was injected, and charge also ejects along that, or propagates along that pili. But here, where they purposely broke the pili, there's no charge propagation. So this is indicative, again, of an electron charge distribution similar to that in carbon nanotubes, and the charge densities are very comparable. So here's one of the controls that was done. For example, if you inject charge into that arrow 5 pili, there's no charge propagation. So the question is, how do the aromatic amino acids get close enough to actually get these overlapping pi-pi orbitals? We don't know exactly yet. We recently begun looking at the structure with cryo-EM, and the images look good, and I think we're going to be able to solve the structure for the pili. So right now what we have is various modeling efforts, and one strategy is homology modeling. I'm showing the results here of how it's thought that the pili monomer comes together to form the filament at the top, and then at the bottom here, the shown in yellow is the distribution of the aromatic amino acids, showing a clear, at least model, theoretical route for electron transport through the pillars. We've tried other types of modeling as well. Just, for example, theoretical model where you just start with first principles with modeling techniques that have successfully recreated the pili of other microorganisms. And when you use the same modeling technique for Geobacter, you get the model shown on the right, which shows even tighter packing of the aromatic amino acids. So it looks like it's certainly possible, and the cryo-EM, I think, will solve the structural questions in more detail. So another important point, which I've already mentioned several times, is these E-pili are decorated with C-type cytochromes, and they're really important. And this was first observed in studies by Qing Liang, who was a postdoc in my lab. And here's immunogold labeling, again showing what I've already referred to before. So immunogold labeling of OMCS on the pili. At first we thought, oh, well, it's just the OMCS is how electrons hop along the pili. That turned out not to be the case. Clearly, these OMCS molecules are separated too far apart. You have to, as I mentioned before, be less than 2 nanometers. These clusters are more than like 100 to 200 nanometers. And the question was, well, maybe the immunogold labeling is not efficient. But when Mikkel looked at this with atomic force microscopy, so it involves no staining or any other approach that might lead to artifacts, you could clearly see that the OMCS was too far separated. But the OMCS is important for extracellular electron transfer. It's one of the most highly upregulated genes when Geobacter is growing on iron oxide, and if you delete that gene, it no longer reduces iron oxide. So here's what we think is probably going on. This is electrostatic force microscopy again. Now, in this case, it's showing dark when the charge propagates. It's just another way to visualize it. So on the left is before charge insertion. Then on the right is when the charge is injected into, this case, the pili, as shown by the white line. And you can see that charge propagates not only all along the pili, but also into this OMCS cytochrome that is attached. So we think that the charge propagates along the pili to OMCS. And in some ways, Geobacter really, it doesn't always have to be OMCS. For example, we published last year that you could, in mutants that lack OMCS, basically recover the mutant phenotype or the wild type phenotype by putting in nanomagnetite. That magnetite attaches to the pili, and the cells function just as well in extracellular electron transfer without the S, or you can actually even just pull, we've pulled sequences out of the environment and express them in Geobacter self-reducens. And if you express the right one, that cytochrome will attach to the pili, and the cells work just fine. So this is our model for iron oxide reduction. So electrons that are generated from central metabolism inside the cell move out along the electrically conductive pili. But the OMCS is important because it helps facilitate electron transfer from the pili to the iron oxide. Now, of course, it's well known that Geobacter species can also exchange electrons with electrodes, either producing current, or if you dial down the potential, you can feed electrons to microorganisms, and get, like Geobacter, and get them to reduce typical anaerobic substrates. like fumarate nitrate, or for other organisms, even carbon dioxide. And some recent studies by Amelia Rotaro suggested that although we always thought about the capacity for microorganisms to use electrodes, which aren't really present in most environments, was due to their ability to reduce iron oxide, there actually seems to be a better correlation with their ability to participate in diet. So that actually may explain some of their good current production capabilities. And when these organisms are producing current under optimal conditions, they can make incredibly thick, electrically conductive biofilms. So this is a biofilm actually from a genetically engineered organism. This biofilm is so cohesive we could peel it off the electrode, so the anode was down here at the bottom. But just like wild type biofilms, this whole biofilm is electrically conductive, and these cells, even at the top here, are metabolically active and contributing to current production. And this is because this biofilm has a conductivity that's comparable to that of synthetic conducting polymers. And this is basically the model for how this works on an electrode. Is the long-range electron transport is through pili. I think the OMCS or other outer surface cytochromes can help with the pili-to-pili electrical connections, but it's absolutely essential for electron transfer from, at the short range, from the biofilm to the electrode to have the participation of C-type cytochromes, and under many conditions, this is a multi-heme C-type cytochrome known as OMCC. So this model has actually been around for some time. There are 2 papers came out last week giving, I guess, basically more confirmation to this concept. And evidence supporting the concept. Remember, the Arrow5 strain produces poorly conductive pili. It can't produce the high current densities because it can't produce an electrically conductive biofilm. And every way you try to improve current production, it's always about the pili. If you knock out most of the abundant C-type cytochromes from Geobacter, you'll actually get biofilms that are more conductive and produce higher current densities because Geobacter starts making more pili to compensate. Or, for example, we used adaptive evolution to select for a strain for high current densities, the strain KN400. This produces a lot more pili, produces fewer cytochromes, but you get much higher biofilm conductance and the higher current outputs. Once you know pili are important for the long-range electron conduction, it's simple to design strains through genetic manipulation. For example, this is strain CL1, and on the left here is that biofilm I showed earlier. It's so cohesive, you could just peel it off the electrodes. And this strain produces biofilms that are much more electrically conductive and produce higher current densities. So we can ask then, well, do other microorganisms have electrically conductive pili? And the answer is yes, some do, and we'll talk about that at the ISME meeting in a couple of weeks. But for this specific type of electrically conductive pili that Geobacter produces, this we've recently realized is due to a relatively recent mutation that occurred in microorganisms closely related to Geobacter. So, and that's, I think, obvious from this image, where we're looking at the structure of the pili of Pseudomonas aeruginosa, microorganisms whose type 4 pili have been studied in great detail. And hopefully you can see that the N-terminal region of the pili molecule is highly conserved in both of these microorganisms. What's different is the carboxyl end. So Pseudomonas has this big carboxyl head, and this is basically missing in the pili of Geobacter. So the pili has been highly truncated in Geobacter. And when you look at the phylogeny of type 4 pili in various microorganisms, you can see that these E. pili that are analogous or homologous to the Geobacter sulfur-reducens pili are really almost exclusively found in phylogenetically closely related organisms. So finally, back to diet. That was a long diversion on pili, but I think it was important to explain that because it is this network of pili that's connecting the cells that's really important for direct interspecies electron transfer in this defined co-culture. And you can look at the references here that show there's just extensive evidence for this. Another important aspect of the diet in that defined co-culture is that we could measure the electrically conductivity of the whole aggregate. And it was more than sufficient to account for the rates of metabolism we see. And I think as and I'll mention this later, diet is starting to be invoked in other anaerobic communities. I think it's really important to have some physical measurement of the rates of electron transport to verify, in fact, that diet is taking place. So as we were working on these geobacter aggregates, and this is scanning electron microscopy, we noticed that their morphology was a lot like aggregates that are recovered from anaerobic wastewater digesters converting various organic waste to methane. So we're really interested in this, of course, and started looking at various anaerobic digesters treating brewery waste. These are Anheuser-Busch digesters converting brewery waste to methane. And from metagenomic analysis, we could see that there was a really high abundance of geobacter. And I was surprised by this. But actually, when you look back in the literature, this had been seen before, but never really mentioned. It was usually said, well, the abundance of delta proteobacteria or something like that. And I think the issue was that nobody really could understand what was a geobacter doing in high abundance in an anaerobic digester where, of course, there's no iron oxide. The other big player in these anaerobic digester aggregates is the methanogen in the genus Methanosata. They're by far the most abundant methanogen, and they are an acetate utilizing methanogen, or that's what they were known for. When you look at the transcriptomics, we see that geobacter are not only abundant, but they're highly expressing their various metabolic gene and account for a vast proportion of the transcripts in the digester. And Methanosata is also metabolically active, accounting for the vast majority of the methanogen transcripts in that system. It was very interesting that by abundance or transcriptomics, there were very few hydrogen-utilizing methanogens. So this is surprising because if these anaerobic digesters go by the standard Bryant et al. Model of interspecies hydrogen transfer, there should be a lot of hydrogen-utilizing methanogens to pull that hydrogen out of the system and keep the whole process going. But you could use fish, for example, and other analysis to confirm that it was really in abundance. It was geobacter Methanosata were the big players in these aggregates. And one of the interesting early observations was that the aggregates had conductivity. In fact, these have a temperature dependence similar to that expected for metallic-like conductivity. And there's a direct correlation between the abundance of geobacter and the conductivity of the aggregate. So this suggested there might be something going on with a direct interspecies electron transfer. And the other evidence or suggestion for that was that Methanosata was a dominant methanogen. So Methanosata is well known and been studied for many years for its ability to use acetate. It was thought to be an acetate utilizing specialist, doing very well with that as a substrate. But as we discussed before, you're metabolizing ethanol, which is one of the big products in brewery waste, you produce not only acetate, but also there's these electrons that have to be dealt with. So the idea was, well, Methanosata is using electrons, but it's not known to use hydrogen or formate. Is there any other way for it to consume electrons? And when you look at the genome of Methanosata, it in fact has the full, complete pathway for reducing carbon dioxide to methane. And in the metatranscriptomic analysis of the digester aggregates, we could see that these genes encoding enzymes in the carbon dioxide reduction pathway were very highly expressed. So it looked like it was reducing carbon dioxide. So we set up a defined co-culture system to investigate this further with geobacter metalloreducens, which I've already told you can utilize ethanol. It does not produce hydrogen or formate. And Methanosata harendaceae, which is a digester isolate, which, like other Methanosata, cannot use hydrogen or formate as an electron donor. Yet when Amelia Rotara put these two organisms together, they grew. And as you can see this, the aggregates over here on the left, visually in the middle is the phase contrast, and here's the fish image. So they're forming tight aggregates of the two microorganisms, and they're very effectively converting the ethanol to methane stoichiometrically with good electron recovery. So this means that the Methanosata has to somehow be using all these electrons that are released during the ethanol metabolism. And it clearly was using them because radio-labeled studies confirmed that radiolabeled carbon dioxide was being reduced to methane in agreement with the expected stoichiometry. So, and if you look in the defined co-cultures and in the digester community, you see basically similar transcriptional patterns, which all suggest that geobacter is metabolizing the ethanol, of course. It's producing a lot of pili, and the methanosata is utilizing not only the acetate, but also the carbon dioxide that it receives from, or that it reduces, using electrons it receives from geobacter. So I'd like to I wish I knew how methanosata directly accepts electrons. That's not known. That's something we're working on. We do know that the electron transfer from geobacter, again, requires the electrically conductive pili, as shown here. You just can't get the co-cultures to work with a pili deficient strain. Now, to date, only one other methanogen is known to function via diet, and that is Methanosarcina barkeri, which is relatively closely related to methanosata. It's also an acetate-utilizing microorganism. Unlike methanosata, it can also use hydrogen. And Amelia was able to get very good co-cultures of the methanosata, the Methanosarcinus, that's these coccoids here, and geobacter growing in this system. More recently, we've expanded the study of diet beyond ethanol to look at important intermediates in a wide diversity of anaerobic environments. So these would be short-chain fatty acids like butyrate, propionate, and even acetate can be metabolized by this diet mechanism. So obviously, diet takes place in at least a few environments. Interspecies hydrogen transfer has been documented in many environments. They're trying to understand what might be the advantage of one process over another. So I think advantage of diet is just faster. So in both cases, you have a syntroph metabolizing ethanol, but once the syntroph metabolizes it with diet, you get really fast electron transfer to the methanogen, whereas diffusion for interspecies hydrogen transfer is much slower. So that's one potential advantage of diet. Diet also, though, has its costs, and I like to think of diet as being analogous to as civilization brought in electrification. So this was a major advance, of course, in how we obtain our energy. Showing on the left, I believe this is a coal delivery, obviously much slower than delivering electricity through power lines. But in order to get that electricity nice and fast, you have to make a big investment in infrastructure to have the power grid to get it to your house. So I think this is the trade-off with diet, is the cells have to invest in the electrically conductive pili and also whatever connectors the methanogens might produce. So, but once you know about this diet process, you can actually get around some of that energetic investment by providing electrically conductive materials to the microbial community. So we first saw this, for example, with granulated activated carbon, abbreviated GAC here, which has been added to anaerobic digesters for years because the idea was it absorbs toxic compounds and it also provides a surface for cell attachment. But it's also highly electrically conductive. And in the presence of GAC, you can get really rapid direct interspecies electron transfer where the electron donating organisms inject electrons into the GAC, the methanogens pull electrons out of the GAC, and under these conditions, even mutants that don't produce the electrically conductive pili or the OMCS cytochrome still will effectively participate in diet. And a variety of materials work similarly with similar effectiveness. So, for example, there's a lot of interest in biochar as a renewable material, and it's also electrically conductive. And what I'm showing here is geobacter, and in this case, Methanosarcina, plugged into the biochar, in directly exchanging electrons through that conductive material. And we've identified a variety of different materials and different strategies for using those materials in anaerobic digesters to help speed up or stabilize that process. Now, another mechanism for promoting interspecies electron transfer are soluble electron shuttles. This was documented by Jessica Smith, who was a PhD student in my laboratory. But it's important to recognize that so far electron shuttles only seem to work with, don't work with methanogenic communities. And I think the reason for this is that the potential of these, at least the shuttles that have been investigated, is not low enough to drive methanogenesis. So that's something that may need some further investigation. Okay, so of course we're interested beyond anaerobic digesters and what is the potential of diet to global methane production. So I stole these images off the web. If any of you are in these pictures, please don't sue me. That was I didn't have any good pictures. But my important point here is that in these terrestrial environments, methanosata are probably the biggest contributor to methane production in terrestrial wetland environments. They've been called the most prodigious methanogens on Earth because they're very effective in acetate metabolism. Now, of course, we know the methanosata are also capable of diet. It helps them grow faster. I think this increases their contribution. And recently, Don Holmes in my lab has, through meta-omics type of studies, demonstrated that the methanosata are participating in diet, and also there's abundance of E-pili expression. So I think this diet is probably happening in some terrestrial environments, for example, rice paddies, which we focused on, and further investigation of that is ongoing. And then the other possibility that has been brought up recently, well, actually Zara brought it up in her original paper, but then has been followed up on, is this possibility of diet in consortia that are anaerobically oxidizing methane. So if you look in the center here, these aggregates that are seen that are oxidizing methane with the reduction of sulfate look a lot like the other images I've already shown of aggregates. And it was suggested some time ago that probably soluble intermediates like hydrogen or formate that would need to diffuse through these aggregates would not be able to account for the rates of methane oxidation that were observed. So really interesting paper then came out last fall on this thermophilic enrichment that was oxidizing methane by Wegener et al., where they were able to rule out the possibility of hydrogen as an intermediate, as interspecies electron carrier, and they saw there was really high expression of pili in this system, and you can see in these pictures from their paper the abundance of pili in the aggregates. My only question about this system is that the pili have conductivity is yet to be verified, and in fact we'll be showing you at the ISME meeting in a couple of weeks. Our studies on these pili suggest that they have in fact very poor conductivity. So, but further investigation is probably required. And then at the same time, this other system was reported, and this is a little bit different. So this is the McGlynn et al. Paper where they suggested from the arrangement of cells that clearly it had to be a conductive aggregate for exchange of electrons. Now the conductivity of the aggregate was not actually measured. I think that would be important to do. But this is a different proposal. And here you can see that the cells are really tightly packed together, and what was found was there's an abundance of outer surface cytochromes. So in this, the concept here is that pili are not involved in an interspecies electron transfer, and it's just cytochrome to cytochrome electron exchange through this aggregate. So I've already told you with the Geobacter system we can't get just cytochrome to cytochrome electron exchange to work. So I think there needs to be at some point a model system to verify this, or at least measure the conductivity of the actual these aggregates to define this. So we've been looking at all this now in more detail. I'm not going to go into that. So if you go into the ISME meeting, David Walker is presenting a poster, and I'm going to talk about this also, where we've been looking further at these conductive filaments in methanogenic sediments from metatranscriptomic data from ANMI and from microorganisms closely related to cable bacteria. And I think there's just a really much more broad story that David Walker has been working out, and we'll discuss more in the future. And then I just wanted to finish up with this concept that I'm also just really interested in, which has been kind of a spin-off of the studies of diet. And this is the idea of the electrically conductive pili representing a novel renewable electronic material. As I mentioned, we recently published that we can genetically modify these pili to, for example, increase their conductivity. We can also change other properties on the pili and how they interact with different materials. So I like this concept because we've developed a strain to mass produce the pili, and it's a really green synthesis from renewable cheap energy sources. For most other abiological conductive materials, even when they're organics, it takes harsh chemical processes, or you'll find toxic components in the final products. We don't have that with the BioWire. They're highly durable, even at, as I already showed you, low pH. They work well in water, and we can modify their properties and tune the conductivity up or tune the conductivity down. We can put winkers on them so they'll bind metals and other materials. And I just think there's a lot of future in this study, and this is just one of the images from our paper that came out a couple of weeks ago, showing how we increased the substantially the conduct. I would have been happy if we doubled the conductivity. First try we got up to 5,000-fold increase, so we're at a conductivity of a kilosiemens per centimeter, which, to put in context, is rivals that of carbon nanotubes. So in summary, I apologize for the glitch I had earlier with the computer, and that I just want to point out that diet or direct interspecies electron transfer is an alternative to interspecies hydrogen transfer. It's been detected in some environments. I think the question is, what environments and when is this important? Clearly, in some of these systems, electrically conductive pili are a route for diet, and the pili just in and of themselves are interesting. This is a novel concept of long-range electron transport, as is the concept of metallic-like conductivity, which I think is leading us into this new field of synthetic conducting electronic materials, and where I think a lot of the focus of our research will be in the future. So I thank you for your attention, and I'd be happy to take any questions.
Okay, thank you, Derek. That was nice. What a cool survey of a wide variety of scenarios for direct interspecies electron transfer. Let's see. Are there any questions from inside the Hangout? And Derek, if you could stop sharing your screen, that would be great, so we can see you. While we're waiting to see if people get their questions together, I have one. I like your analogy with the power grid, the electrical infrastructure building. It raises the question: when does Geobacter decide to invest in something like that? Do you have any sense of what kind of ecological conditions force the decision to go that way as opposed to not, or what are the circumstances where that makes them more competitive?
Sure. So it basically does it when it feels like it's drowning. When it doesn't, if it has a soluble electron acceptor, it doesn't express pili. Even a chelated iron or electron shuttle, if it has that, it doesn't need the pili. But as soon as electrons start to build up in the cell, and I don't know how it senses it, but it really tightly regulates that, and now it'll express the pili because it's got to get electrons outside the cell in order to survive.
Yeah. Okay. So it's you think it's responding to an actual redox condition internally, potentially?
Yeah, that would be my guess. I mean, it's certainly, that's the environmental signal. So it's like, oh my God, I can't. There's nothing for me to dump electrons on. I'll put out these pili and see what I can find.
Yeah.
I mean, that's my simple-minded way of thinking about it. So.
Yeah. Yeah. Do you have any sense of the energetic cost for doing that? I mean, have you measured or theorized about how much ATP it takes to construct all these pili or something along those lines?
Yeah, it definitely costs something, which is why the cell doesn't do it unless it has to. I mean, I can't tell you off the top of my head what the percentage of its overall net energy yield or cell biosynthesis goes to the pili. I mean, of the overall biomass, there's still a fairly small proportion, but it's a significant investment. Otherwise, it wouldn't bother to regulate.
Right. Yeah. Okay. Do we have other questions? Anybody inside the seminar? Anybody on Twitter? We got a couple comments on Twitter. Yeah, Leanne Benning had a question, but you answered it. Let's see. Anything else for Derek? Got him for a couple more minutes. Okay, so it sounds like you're going to be speaking at ISME as well. Is that right?
Right.
Okay, and is that next week? When is that?
I hope it's not. No, I think it's the week after next.
Okay.
to check.
Great.
Yeah.
Okay. And then if people want to get a hold of you after the fact, or let's say they're watching this sometime in the future as it's recorded, what's the best way for them to ask you questions?
I think just shoot me an email. Probably is the best. Yep.
Perfect. All right. And Derek is also on Twitter at Microbelectric. And we thank you very much for your time. We really appreciate it, Derek.
Thanks, Cameron. It was a real pleasure. Thank you.